After Avery's experiments, scientists still did not know what part of the cell, DNA, or protein was the genetic material. Alfred Hershey and Martha Chase conducted experiments in 1952 to discover where the genetic material is found. They used bacteriophages to figure this out because bacteriophages are made of only DNA and a protein coat. They wanted to discover what part of the virus, called a bacteriophage, contained the genetic material by seeing what part of the virus entered the bacteria. This is a diagram of a bacteriophage. In here is the DNA, and this is in the head, which is this entire part. What's called the tail sheath, this part, and these are the tail fibers, and around everything is the protein coat.
They used phosphorus and sulfur as markers, seeing as protein contains almost no phosphorus and DNA contains almost no sulfur. Therefore, they put phosphorus 32 in the DNA and sulfur 35 in the protein coat. If the DNA entered the cell, they would be able to notice the radioactivity of the phosphorus. If the protein coat entered the cell, they would be able to notice the radioactivity of the sulfur. These are two different experiments. The first one is when the bacteriophage had phosphorus 32, this is the phosphorus 32 in the DNA, the bacteriophage infected the bacterium, and when it left, they saw that there was radioactivity inside the bacterium, meaning that the DNA had been injected into the bacterium. The experiment was when the bacteriophage had sulfur 35 in the protein coat. The bacteriophage infected the bacterium, and when it left, there was no radioactivity inside the bacterium. The DNA had entered the bacterium, but the radioactive protein coat had not.
Their discoveries showed that the DNA is what entered the bacteria, not the protein. This confirmed Avery's results that DNA is what contains genetic information.